Bismillahirrahmanirrahim. Assalamu alaikum to all our viewers. This is our educational channel Learning Biology Online and our third lecture on cell, where we will discuss different important components within the cellular environment, starting off with the cytoplasm. Then we have endoplasmic reticulum, ribosomes, and lastly the Golgi apparatus. So we will be focusing on their structure and their functions that are performed within the cellular environment. So by the end of this lecture, we hope that you will be having a very good idea of all these organelles, their structural component, and how they functions within a cell. So starting off with the definition of the cytoplasm, where we have an aqueous ground substance where different organelles are present, and we can also see different waste and other metabolic activities occurring within this area. So this is how we define a cytoplasm that consists of an aqueous substance and different organelles are present within it, और different sort of chemicals भी हमें इसमें मिल सकते हैं due to massive metabolic activity that is occurring within this environment. Coming to the component, the soluble part is known as the cytosol. Soluble part means कि जो भी metabolic ions हैं, they are basically dissolved in the fluid components. So you can see the different sort of ions and proteins that are present within, and the colloidal solution component. अब colloidal solution component का मकसद ये होता है कि जो different macromolecules हैं, they are basically not dissolved in the aqueous environment over there, जिसकी वजह से हमें जो different ions हैं वो नज़र आते हैं floating within the cytoplasm under high magnification. ठीक है. सो इलेक्ट्रॉन माइक्रोस्कोप में हमें ये डिफरेंट फीचर्स जो है नज़र आ सकते हैं. तो क्लोइडल सोल्यूशन का बेसिकली हमें आइडिया होना चाहिए कि इट्स अ सोल्यूशन इन विच द पार्टिकल्स ऑफ द मिक्सचर दे आर नॉट प्रॉपरली dissolved, so that is why it may appear as sol or a gel component. Gel is something non-viscous and sol is the soluble component. So this is how the colloidal solution within the cytoplasm may appear. So as we have already discussed कि macromolecules क्यों present होते हैं, because of presence of high metabolic activity. So we know we know cytoplasm as a storehouse of the vital chemical reactions, क्योंकि जितने भी chemical reactions हो रहे हैं within the cell, they require an aqueous environment that is provided by the cytoplasm. So different जो reactions होंगे, उनका जो waste बनेगा या जो भी product बनेगा, that is also present within the cytoplasm. तो एक बहुत ही इम्पॉर्टेंट प्रोसेस जो अगर कर रहा होता है ग्लाइकोलिसिस का, दैट इज़ सोर्स ऑफ ग्लूकोज टू आवर सेल, दैट इज़ आल्सो अकरिंग within this environment that we have mentioned over here for the sake of example. Coming to endoplasmic reticulum, that is found as a network of channels around the nucleus within the cytoplasmic environment. And then a very important component of the endoplasmic reticulum are the cisternae, that are spherical or tubular structures through which exchange of different material within different organelles that can occur. So here we can see the diagrammatic component of the rough endoplasmic reticulum. You can see how much close it is to the nuclear membrane, that is the covering of the nucleus. And then you can see the different granules that are attached to the surface of the endoplasmic reticulum. So this is the spherical component, ठीक है, जिसके ऊपर आपको डिफरेंट राइबोसोम्स अटैच नज़र आ रहे हैं. तो ये बेसिकली वो राइबोसोम्स हैं जो न्यूक्लियस में ही बनते हैं. थ्रू न्यूक्लियर पोर जो है, दे कम आउट इनटू द साइटोप्लाज्म एंड दे मे अटैच टू द सरफेस ऑफ एंडोप्लाज्मिक रेटिकुलम, or they may be free within the cytoplasmic environment. Then you can see the smooth endoplasmic reticulum and its tubular structure. तो यहाँ पे आप देख सकते हैं कि जिस endoplasmic reticulum के पोर्शन पे ribosomes attached नहीं हैं, they are mentioned as smooth endoplasmic reticulum. Coming to the comparison of these two different types of endoplasmic reticulum, the smooth one is involved in detoxification of any waste substance that could be present within the cell, and then you can see the transport and its property for mechanical support of the cell. Finally, we have the rough endoplasmic reticulum in comparison, which is involved in synthesis of different sorts of proteins that may stay in the cytoplasm or may be exported out of the cell. Then coming to the ribosomes, that are tiny granules which may be found attached or dispersed within the cell, and they are mostly attached to the rough endoplasmic reticulum or dispersed within the cytoplasm. So these are the two different states in which you can see the ribosomes. So these were discovered somewhere in the 1950s by scientist George Palade, who also found out that these ribosomes, they basically develop within the nucleus, and there is a central dark portion of the nucleus known as the nucleolus, where these ribosomes are formed, and then they are transported from that component through the nuclear membrane onto the surface of endoplasmic reticulum and into the cytoplasm as well. So this is the origin of these structures. Then you can see how they develop from smaller subunits that are known as the polysomes. So each eukaryotic ribosome consists of two subunits, and they have a very important role in formation of different sorts of proteins. You can see how the two different subunits are involved in formation of ribosome, both in case of prokaryotic and eukaryotic cell. So you can see how their size is also going to vary. The eukaryotic ribosome, that is formed after combination of 60 and 40 subunit, and the final product is the 80S, that is larger in size as compared to the 70S in case of the prokaryotic cell. Coming to Golgi apparatus, that were first discovered in 1898 by budding off from the smooth endoplasmic reticulum. So they appear as stalks of flattened membrane bounded sacs. ठीक है, so pouch like इनकी appearance होती है, and those pouches or sac like substances, they are very close to each other and they are interconnected through tubules. So these sac-like structures that are formed from the rough endoplasmic reticulum, that are known as cisterna. So as we have already discussed, that those stalk-like sacs, they are interconnected with each other, and their convex surface is known as the forming phase, whereas the concave inner surface is known as the maturing phase. So if we want to understand this structure diagrammatically, so this is how you can see it under the microscope, that you can see different vesicles from the endoplasmic reticulum. They are budding out from here, and this component of Golgi apparatus basically develops from the smooth endoplasmic reticulum. So this convex surface is known as the forming phase, and the inner, that is surface, that is concave, it is the maturing phase. So this probably would be the case कि ये जो इसका outer surface है, it is involved in formation of vesicles, और उनकी जो फिर modification या maturation जो है, that occurs within this inner surface, that is concave. And you can see this stalk like structure as a whole, it is known as cisterna over here. So this is the cis phase that you can see over here, that is closer to the endoplasmic reticulum, and the area that is away from the endoplasmic reticulum, that is known as the trans phase. So you can see how the vesicles, they are formed over here, and then Golgi apparatus is basically involved in transport of different sorts of materials that are present within the cell. So this is how the whole cisterna associated with the vesicle, this whole structure is known as the Golgi complex, and you can see how it functions. It is involved in basically the modification of proteins that are formed in the ribosomes. They are modified in the Golgi apparatus, and then they are transformed into vesicles for their secretion from one environment to another, that is from the cell to the external environment. So this is the function of Golgi apparatus, that it is involved in modification of protein and other molecules, and then it transforms them into vesicles and it sends it to the external environment outside the cell. So do like our video and share it with your colleagues if you think it can be helpful for them in understanding all the topics mentioned in this video, and in the comment section do ask if you have any question relevant to these topics. Finally, do not forget to subscribe to our channel and click the bell button so you can receive the updated uh, lectures of other topics on regular basis. Thank you very much.